Good morning, everybody. Hopefully you had a really fantastic evening last night. It was absolutely wonderful to be in Paris and be on the boat on the river. It really was magical, I think. I think we all very much enjoyed that, and it was a wonderful finale to the One Health EJP. So this morning I have the pleasure of chairing the first session, and I apologise for the slight delay. We had some technical issues, but we will try and catch up time as best we can. And the first session today, we're looking at the One Health EJP contribution to science, health policy and society. And in particular, we're going to be focusing on impact on science and how the One Health EJP has particularly contributed. And we've heard so much around this area from a perspective of outbreak preparedness, publications that we produced, driving policy change, collaboration, particularly the multidisciplinary collaborations, and dissemination and exploitation throughout the project. So we've got 4 speakers today who are going to be talking to us about different aspects of how we've impacted on science. And it's my pleasure to start off with our first speaker, who really doesn't require an introduction, but everybody is aware that Hein Imberechts is the coordinator of the One Health EJP. He's a vet background and has a PhD and scientific background. Currently works as support advisor at Sciensano in Brussels, and he's the scientific coordinator of the entire One Health EJP and vice president of the MedVetNet Association. He also coordinates the SCAR Collaborative Working Group on Animal Health and Welfare, and as such is involved in the planning of the European Partnership on Animal Health and Welfare, which is just starting. So with no further ado, it's my pleasure to invite up Hein.
Good morning, everybody. It's really a pleasure to be here, and after this wonderful evening we had yesterday. So thank you very much for the organizers. Let's move to the next. Well, the talk of today is to give you a little bit of a flavor of what we have done, and we is all the EJPs, all the collaborators, all the scientists, all the experts that have been working in the projects. I will be focusing on sorry, the research projects. PhDs are very much involved in this as well, and touch upon the GIPs as well. I think what we all together have realized is generated a lot of evidence, of new knowledge, and we have created this networking. I'm really much convinced that the main achievement of this One Health EJP is its networking, and I want to stress that again. And for the GRPs, we organize these internal calls. So it's not like an internet external, but internal among the partners. And it's a little bit interesting to say we organize that according to what we call the themes: detection, host pathogen interaction, epidemiology, risk assessment, and response. And it is interesting to see that all the projects that have been developed, that have been started, launched in the EJP, touched upon all but one of these themes: host pathogen interaction. I'm talking now for the GRPs, not for the PhDs, but for GRPs. There was not one project that was proposed, which means that scientists already and the scientific world within the EJP was aware that the EJP is not a regular research project, but it's about the laboratory working together. It's about methodologies. It's about knowing each other better, aligning the capacity of the different institutions. And I use again this slide, these different steps of the process towards prevention, detection, response, or preparedness. The different steps. The first step is about surveillance. We need good surveillance systems in order to have good data, we need good laboratory, we have to do this joint risk assessment, and then there is this risk management. So the different steps towards, and this is really important, and we called it from the beginning in 2016 or something like that, we already put that forward, and this is our interpretation, because this is multidisciplinary approach, and it's something that has to be done in the animal health, in the public health, and the food safety. So this is our clearly, each time that I tell this, and I tell this many times, this is for me our model for One Health. This is, and it worked. It worked. So this is really important to show. But we came up with so many results, far more than we can explain here, and therefore we decided to have this kind of overview table of what is in there in the EJP. And this slide and the next one shows in the second column these different steps that I showed you in the circles in the previous slide. So the different steps towards the preparedness. And all the GIPs on the left-hand side, they can be aligned to that backbone. But also the GRPs, the research projects, all the outputs and our outcomes can be aligned as well. So that means that all these projects that we have set up, these internal projects with all the 44 partners, means that they all contributed to the One Healthness of the partners. So this is the main message of these slides. Okay, scientifically, I think we did well. I don't know, I cannot interpret. We have 265 publications altogether. Of course, there are a lot of projects, and there are still publications to come. I think this is nice, certainly. We know that there are, and I think Peter Jan, you said that you're starting, and I heard from others as well, that you're starting new projects based on the results of this. So there is an impact on science of these projects. But I think what is really important, I stressed it already, is this cross-sector, cross-border collaboration. This is really important. This is a foundation for the future. We are building trust. We said it yesterday, so important. And of course, trust is needed when it comes to an outbreak, that we have to work together with our peers abroad as well, with the other sectors as well, so that we can work this out. It is very important to stress that researchers, they do their job. Their job is looking at the proposal that they have developed and delivering on what they propose to do, but then their job more or less stops, and then others have to take over to use these results to turn them into outcomes that can be taken up by interested parties, be it in science, be it for society, the authorities, be it for the POC members, and so on, but for international stakeholders. So this is really important as well, and there are other needs, and that's where communication comes in. We have the results, that's one thing, but the effect of the results, the outcomes, the long-term effects, the impact, that's something that others have to do, and communication is key to that, and that's the role of communication as a first step in dissemination and exploitation. What I can say, we in these GRPs, there were a lot of, well, 3 domains, you know, we always used the wording domains for foodborne zoonoses, AMR, emerging threats, and there were so many pathogens that all these researchers worked on. I don't run through this list. And in very multidisciplinary work on free extracellular DNA to know where how resistance genes, for instance, can spread. It's an interesting thing is also, for instance, the use of food purchase data as a proxy for exposure of people. So interesting approach. There's a lot of technology behind in all these different projects and so on. I cannot go into detail, but it's clear that there is a very broad area of results that have delivered, and all the results, they fit into this prevention detection response. So they all contribute to the impact of the EJP. Due to the sake of time, I will skip a number of slides. It is okay. Maybe what is better what I can I can refer to what we have said yesterday. In the second call for GRPs, we had in the 3 domains, foodborne zoonosis, emerging threat, antimicrobial resistance, there were a number of projects that have been started, have been launched. And yesterday we said that the EFSA and ECDC in the beginning 2018 helped us in identifying the needs. And you see well, it's an internal document, but you see these SH stakeholders, they helped in identifying these needs. And just to mention one on the top, Pharmet and WorldCom, they responded to a need of identifying those stakeholders on-site tests, rapid on-site tests. So there was a request of EFSA and ECDC. Also down the line, the penultimate one, Adonis. Well, you know that Salmonella enteritis is a pathogen which was very high in the beginning of the 90s, 2000, and then it diminished, its prevalence diminished in human health, and then it stopped decreasing trend. And EFSA really wanted to know what is at the origin of that. And so that was also another project that was started. I just mentioned the parasitology projects. There were 3, and they created this network of parasitology that really feed into the request of WHO and the European Reference Laboratory of Parasitology, EFSA, ECDC. So really impactful projects were started. So what I can say to wrap up, I think we organized this network, which is really important, and also focusing on not only generally in One Health, but also focusing on a number of domains. I mentioned here foodborne zoonosis, antimicrobial resistance, parasitology, but there's also on technology and so on. So there are a lot of networks that were created, very good. And there is a lot of information. It's so difficult to show you and with you, to share with you all these results. Please have a look, and Jenny will be pleased that I say that. Please have a look at the website, because really it is functioning very well, also with the search function. So please have a look. And my final message is, it will not stop here. We will stop the meeting within a few hours. The EJP will fade away, but it will not. It will still linger on. It will be still there. It will have another name. It will be the MedVetNet Association, and I'm really looking forward to seeing what would happen over there. So thank you very much for your attention, and I give you the word.
Thank you, Hein. Maybe you could join me on the stage here, and in the interest of time, we'll have questions at the end of the session. So our next speaker is going to be speaking to us particularly about a research project in the One Health EJP, and we're going to be looking at the BeONE project, and Verónica Pinho Mixão is going to speak to us about that project. Verónica is a postdoctoral fellow of the One Health EJP BeONE project, currently working specifically as a researcher at the Genomics and Bioinformatics unit at the Portuguese National Institute of Health, INSA, and involved in the establishment of the whole genome sequencing-based surveillance framework for foodborne pathogens. So with no further ado, Verónica, if you'd like to start your presentation.
Okay, so good morning, everyone. First of all, I would like to thank for the opportunity to be here presenting some of the outputs of the... Okay, sorry, I was too far. Some of the outputs of the BeONE project. For those of you who are not familiar with the BeONE project, this project aimed to build integrative tools that enhance evidence-informed public health decision-making, connect European genomic surveillance laboratories, and promote the comparability and innovation in One Health surveillance, always with the focus on foodborne bacterial pathogens. Given the impossibility for the project leaders to be here giving an overall picture of all our outcomes, I'm here to present some of the results of Work Package 1, which is the work package in which I did my postdoctoral research. So genomics-based surveillance for foodborne bacteria is being implemented, or is actually implemented in many European laboratories. Some laboratories more advanced than others, as yesterday was mentioned. But different laboratories run different bioinformatics pipelines for the analysis of genomics data. And this is posing a challenge for the integration and communication of data between the laboratories, because we are never sure how can we compare results that actually were generated by different pipelines, by different methodologies. But actually, are the results so different, or are they congruent and we could actually compare them? So taking the chance that we had this huge network with multiple countries, different sectors, we had some institutes running different types of pipelines, allele-based pipelines, SNP-based pipelines, commercial softwares. We decided to create diverse data sets of important foodborne bacteria, Listeria, Salmonella, E. coli, and Campylobacter jejuni. We provided the BeONE partners with these data sets, and we asked them, please run your surveillance pipelines on these data sets and tell us the clustering results you obtain at all possible resolution levels. So then we can compare them. And here we faced a challenge, which is, okay, if the results are coming from different pipelines, they have different formats. We need to harmonize them somehow to be able to compare them. And we also needed to have clusters at all possible threshold levels, because I'm not sure if you are familiar with that, but many pipelines actually rely in the end on a researcher or on a technician looking at a phylogenetic tree and trying to identify what actually is a cluster. Not every institute has an automated way of doing this. So we developed Report 3, which is a tool that takes multiple input formats, so it's able to deal with a huge variety of pipelines. It automatically identifies the genetic clusters at all possible resolution levels. It integrates metadata that is available, generating automated surveillance-oriented reports. And Report 3 is already implemented and in place at INSA. It has really fastened our surveillance, not only of bacterial, but also viral pathogens, and it plays a key role over there. And it's also being integrated in the cohesive information system of the One Health EJP. So with Report 3, we were able to process all the outputs of the different BeONE pipelines, let's say. And with that, we could do an evaluation of the clustering at all possible threshold levels for each pipeline, do a comparison of their stability regions. I will not enter in much detail into this. They are usually regions used for new nomenclature design. We could also do the assessment of the congruence with traditional typing methods, with known population structure of these species. And also we did pairwise comparisons between the pipelines. And with that, we could have an idea of the congruence between the different pipelines at all possible threshold levels. And currently, we have at hand like some kind of a dictionary of correspondence between the pipelines. And we know what is, if my results at doing with this procedure at this distance between the samples in one pipeline corresponds to this other result in the other pipeline. And of course, it had to be, we did the evaluation of the performance of each pipeline in the identification of potential outbreak-related isolates. This was a huge amount of data. Unfortunately, we did not manage to compile everything in a publication before the end of the One Health EJP program, but all these results will come out very soon, this year, still. And besides all these comparisons with many pipelines, which I call the BeONE pipelines because they are the pipelines of the BeONE partners, we will also release the set of tools that actually we developed to do this cluster congruence analysis. So actually, any laboratory can take these tools, run on their collection of whole genome sequencing data, and not only compare with other laboratories, but also analyze their own pipeline. And we hope with that to facilitate the technological transition, also promote the long-term sustainability of any pipeline, because an institute can see the real impact of a software update or of a schema alteration on the clustering results that they have in their laboratory, so they can do these transitions. And we also hope to promote the establishment of a full integrative and efficient One Health surveillance by facilitating the communication and integration of data between different laboratories. To finalize, I just want to thank publicly to all the BeONE partners for their commitment with this exercise, because during the pandemic run, their own pipeline stopped the surveillance. They didn't stop, but they did in parallel with the surveillance all these analyses. And of course, the One Health EJP for the support of this project. And thank you all for your attention.
Thank you, Verónica, for a fantastic presentation. We'll have questions at the end. So our third speaker is going to be, our third speaker is specifically going to be speaking about the One Health TOX-DETECT project, and Yacine Nia from Anses is going to be the speaker. Yacine is Deputy Head of the Staphylococcus, Bacillus and Clostridium Unit and responsible for Staphylococcus team at Anses. He is also Deputy Head of the European Union Reference Laboratory for coagulase-positive staphylococci and Coordinator of the TOX-DETECT project. Welcome. Welcome, Messi.
Hello, everyone. Good morning. Thanks a lot for giving me this opportunity to present the outcomes of the project TOX-DETECT that was coordinated by Anses with Jacques-Antoine and Hein and myself from 2018 until 2020. So let me to start to give you just a short feedback, a short background. Sorry, short background on these bacterial toxins. So each year, European Union member states report the foodborne outbreaks occurred in their countries to EFSA and ECDC. And if you have a look on this figure, we can see that between over the 3,000 outbreaks reported by EFSA, there is about, it depends each year, 15 to 20% of outbreaks occurred due to bacterial toxins. Bacterial toxins mean toxins produced by Staphylococci, Bacillus, and Clostridium. And if we have a look also on the percentage of each pathogens, you can say that it depends each year, but we have Bacillus cereus as the first source, the second one staphylococcal enterotoxins, and the last one Clostridium. But it changes each year. If you have also a look just on this color, we have the green here represent the strong evidence. What I mean strong evidence that we are sure that this bacteria is the responsible of this outbreak. Weak evidence, so we have a doubt. Why? It's easy, just we have a lack of methods allowing to well characterize the involvement of these pathogens in outbreaks. So for this reason, we proposed the TOX-DETECT project with the main objectives. The first one is the establishment of an EU, European Union network, focusing on the detection and identification of these three pathogens, Staphylococcus aureus, Bacillus cereus, and Clostridium perfringens. The second one is the evaluation of different diagnostic approaches, example, mass spec, immunological diagnostics, and functional approach. Why? Because for detection of bacterial toxins, in general, we use directly ELISA methods, or for detection of pathogens, we use MALDI-TOF using a commercial database, but mass spec was not challenged. But this method can give us a lot of information on proteins, for example, and also immunological tools to try to work on the new reported toxins. We have also as objective generation and characterization of a reference collection of bacterial strains issued from different from One Health concept, I mean environmental strains, food strains, and clinical strains. Implementation and development of methods to identify bacteria and associated toxins and or virulence factors. And finally, once the methods were developed and optimized, the idea is to transfer this method, so to evaluate that this method can be transferred to the different laboratories. So the main results of TOX-DETECT, the first one is to exchange on quality assurance practices, SOP, standard operating protocol, and the materials to promote work on bacteria producing toxins. So this SOP means that when we develop the method, we established one common report. Each one harmonized the drafting of the protocol, and we try to have all information that allow the transferability of this method. So we fully characterize strains, more than 100 strains that are today available for partners, and were also shared with partners of the project. We established, based on these 100 strains, a multi-TOF library that it is available today and was also shared with more than 13 partners and with national reference laboratories, especially for staphylococcal coagulase positive staphylococci. We have 15 standard operating protocols shared on Zenodo and 6 methods from MADITOF, MASPEC, and ELISA developed and are available also. A proof of concept and robustness and transferability by organization of 5 inter-laboratory was organized. So, to summarize, to test that the method can be transferred, we established 5 inter-laboratory tests. Over these outcomes also, we shared the results by different European projects or BioTOX projects, for example, that worked on abrin, ricin, and Clostridium toxins. It was presented also at OPCW and also shared largely with European Union Reference Laboratory for Staphylococcal coagulase positive, as we are coordinator and responsible of this network. But we have one question that remains: how to disseminate developments and results for bacteria and toxins not covered by European Commission regulation, I mean Staphylococcus, Bacillus and Clostridium. Scientific and impact, just 1 minute, it will be okay. Development, so tools developed are development of toolbox strategy, including various complementary principles with direct impact on foodborne outbreaks characterization. We improve consumer protection and by filling gaps of lacking methodologies, especially on non-targeted toxins. For example, for Staphylococcus, we can detect only 5, but we work now on 24 enterotoxins, improve FBO characterization, and we go from weak evidence to strong evidence, and improve also the implementation of Directive 2003/19/9. So I would like to thank OHEJP coordination for their help, and especially during COVID period. And I would like also to thank all partners involved in this project. Thank you.
Thank you, Yacine. If you join us on the stage here. Okay, so our final speaker today in the session is our presentation of the One Health EJP co-financed PhD, which is LIN-RES PhD, and Cécile Boland is going to be speaking online for us today, specifically. And Cécile is head of the unit of general bacteriology in the veterinary bacteriology services at Sciensano. She was the project coordinator and PhD supervisor of the LIN-RES project. So welcome, Cécile.
Do you hear me well?
Yes, we do. Yeah.
Yeah, sorry, I had a loss of connection.
Yeah.
Okay.
We can't see your slides yet. No. Yeah.
Okay. Everything is okay? Sorry. So thank you for giving me the opportunity to present to you the key impact of the LIN-RES PhD project. So next slide, please. Yeah, so first, let me start with the key results and a little of background. So linezolid resistance is critical for human health because linezolid is a critical antibiotic to treat multi-drug resistance bacteria. Through our LIN-RES project, we conducted a linesolid selective monitoring, through which we observed a much broader reservoir in food-producing animals than observable to the classical official monitoring, which is not selective. So we observed the highest rates in veal calves with 16%, and in pigs with 25% of pig samples that carried linesolid resistance bacteria. Moreover, we observed that in most of the collected isolates, in 97% of these isolates, we observed linesolid resistant genes that are known to be transferable. So all of these represent a threat for AMR transmission to humans. And our project highlighted that a cross-selection with the use of florfenicol will maintain linesolid resistant genes. Next, please. Yes, and so the first key input was that we communicate all these highlights through a research paper published in 2021 in the Journal of Antimicrobial Chemotherapy. And the second key output is the PhD thesis defense itself that occurs in 2022. Next, please. So our key results were shared with Belgian institutions, so including the Food Agency for the Safety of the Food Chain, the Medicine Agency, and the AMCRA, which is a knowledge center on antimicrobial use and resistance in animals in Belgium. And we shared also all of this with the European Reference Laboratory on Antimicrobial Resistance, the EFSA, and the European Commission. Based on our outputs, a technical working group was created in Belgium on the question of the use of florfenicol in animals and cross-selection of linesolid resistance to think about the current guidelines. And the last outcome is that we are being settling a European Reference Laboratory Antimicrobial Resistance Working Group to agree on an harmonized protocol to launch soon a voluntary selective linesolid monitoring in several European countries, with the aim to carefully monitor this resistance in animals. Next, please. So the output of this project highlighted the needs for a good surveillance and understanding of the mechanism and spread of resistance in general, and to promote a prudent use of all antibiotics in a One Health perspective. So not only the resistance to, not only the use of critical antibiotics for human health, but really all antibiotics due to this mechanism of cross-selection. Next, please. Our results are publicly available to compare with future monitorings and researches, and so all the genomes are published in NCBI database. Next. Our results raised awareness of the human and animal sector on this issue of linesolid resistance. And next. Last but not least, a reflection and adaptation of the Belgian guidelines on the use of florfenicol in animals is ongoing in Belgium, and with the aim to limit the risk of selection and spread of linesolid resistance bacteria in animals. So thank you all for your attention, and a special thanks to the PhD student, Michael Timmermans, who was the PhD doing all this work, and to all the colleagues and partners which have contributed to the success of this project. And thank you all for your attention, and sorry for the little technical issue that I encountered today.
Thank you, Cécile. A really wonderful presentation, very enjoyable. We are running a little bit late, so we will have to just shorten the question session, but I think we have time for just 2 quick questions, and we have all of the speakers available here. So I'd like to ask the audience if there are any questions at all that they'd like to ask to any of our speakers. Maybe oh, there's one at the front there.
Gilles Salva, Anses. I have a question for Yacine. During the last outbreak we have with Bacillus cereus, do you use the results of this program
Okay, thank you. So you mean the outcomes of this project? Okay. Let's say some or a part of this outcomes were used. For example, the implementation of the PCR and research of gene encoding other toxins responsible for this outbreak. Yes, was implemented, but the method that allow the detection of the toxin emetic, there is a standard. So for the serolide. But yes, genomic analysis and genes and PCR were used. Thanks.
Thank you. Any additional questions? I had a quick one for Verónica, if I may, and really enjoyed your presentation, but just wondered, how do we encourage groups to take on some of the pipelines and ensure that we have everybody using the same pipelines and that they are harmonized in some way?
Okay, so I think that's the $1 million question, right? Well, but okay, so the thing is, honestly, I'm not sure if we will manage to make everyone use the same pipeline, and that's actually the key message of our study, which is, okay, maybe we don't need to use all the same pipeline. We can simply try to integrate our results and make everything compatible. Of course, we will have to agree in guidelines and in the best practices. Everyone will have to follow some rules. But I think with this kind of work and with this kind of tools, we can converge and merge the results from different pipelines. I think that's it.
Thank you, Verónica. Very diplomatic answer. I think in the interest of time, we'll close this session, but I'd like to please join me in thanking all of the speakers once again for wonderful presentations.
Thank you.